Thank you. So, morning everyone. I hope I didn't kill you yesterday with the seminar. So, what we're going to see today is a little bit of how to do what you saw yesterday yourselves. Basically, from all the modeling approaches that we discussed yesterday, I would like to introduce to you today a bunch of them, from Boolean Logic models to differential equations. All of them can be used to model signal transduction and some other molecular processes that you might be interested in. So let me introduce to you what we're going to discuss now. So the first step to use modeling to try to understand your experiments is actually to have a question behind, of course. Then once you have a question, the next step would be that it's crucial to set up the experiments in a way that you will extract the most information. Next step is to process data correctly. As you know, this is something that you have been seeing in the past few days during the course. For example, you have worked with fold changes and some types of normalization. The next step then, once you have processed your data, is to choose the right type of mathematical model that you're going to use to try to understand your question. So this is specific, and you need to know a little bit of the field to make the decision which type of model I'm going to use, given the question, the data, and so forth. Once you have chosen your mathematical model, one way to make them predictive is to train models to experimental data. We're going to see how to do that. And finally, the most controversial point is use modeling to gain insight. So I guess you have heard some of the controversy about using models. Some of you brought that question up yesterday about after the seminar. And this relates to the fact that some people find that the mathematical models are actually far away from reality, or they don't quite represent what they are looking at. And in a way, it's true. So there was this sentence by George Box, which was a famous statistician, who said that most models are wrong, but some are useful. And that's the key to it. So basically, all models are approximations. They represent the way you understand, or the way your hypothesis, basically. As we saw yesterday, for example, with the model of the Earth that was used before people knew it was round. So it's key just to try to not be too ambitious when you try to use the model to understand something. It's good to know the limitations and the assumptions. If you do that, I believe modeling can be very useful, as we will see today. So what we will do is assume that you have a question, and we're going to discuss these 5 points. After that, so once we see how to decide what type of model, how to process your data, and more or less you have an overview of the field, we're going to do 2 practical sessions, 2 hands-on sessions where you're going to do it yourself. The first session is discovery-based. So you're going to take a list of identifiers that can be the peptides or the proteins that you have found from your mass spec experiment, for example. It's discovery-based because you don't assume any prior knowledge. So what you're going to do is you just take this list and try to do some downstream analysis, try to see what networks are regulating your data, which are the processes that are active. Can we enrich this? Can we find functional clusters within this list of identified peptides? That's discovery-based. The second part is going to be prior knowledge-driven. So we're going to do something like I introduced yesterday. We will assume that you have a signaling pathway that you're interested in, or a process, and you already know what your hypothesis is, and then we're going to try to compare that to data and do all of that. So it's 2 hands-on sessions. And so then just logistics, like Eduard said, we're going to have a coffee break at 10:30. So hopefully that will be after this introduction, and it will be a nice break after we have started with the first hands-on session. Cool. So let's see how to set up the experiments to extract the most information. To that end, let me just use the Plateaus allegory of the cave, which is super famous, as you know, and it's just to illustrate that. So as you know, in here, what happened was some people were locked to a wall for the whole of their lives, and they had to interpret reality through some shadows that were projected using a fire and some shapes. So here, what I try to illustrate is that what you're looking at when you do the experiment is actually just the shadows that are projected. It's not normally that you see the reality, really. So you have to take that into account when using later processing of the data. For example, to model signaling, proteomics is the natural source of data. But which platform can you use? Which is the one that best represents, or which is the shadow, let's say, that best represents what you want to see? As you know, proteomic experiments can be very broad or different, both in coverage and in the number of, both in number of samples and number of target proteins or peptides that you can identify. So this is just an overview to see that there's a trade-off between target proteins. So for example, there's approaches that enable to see a lot of target proteins, right? Like for example, mass spec. Whereas there's approaches that are more specific and like flow cytometry, Western blots, and so on. There, the trade-off though is that you are going to have a lot less sampling. Right? So this is something that you have to take into account, and because of this trade-off, then for example you can use like antibody-based approaches more for validation, for example, or mass spec for discovery. Then I would like to use an example of a published paper of a publication recently published to illustrate how to set up the experiments properly. So in here, this is a paper that you have in the materials. It's printed for you. And what we were trying to do was to, by the way, I'm sorry about the resolution. Let's cut here. So what we're trying to do is understand how crosstalk is regulated in yeast. So for example, this could be a yeast cell, and what we were doing is trying to understand what happens when you stress the cells using a high concentration of salt. And then it's known that a pathway that regulates the stress response is mediated by HOG1. And thereby what it happens is that the in and out water balance is controlled. And also, if you put just way too much salt, instead of controlling the in and out water balance to deal with osmotic pressure, the cell, what it does is go into cell cycle arrest. So this is what happens when you put salt, and it's mediated through a signaling cascade mediated by HOG1. If you use a mating pheromone, for example, like an alpha pheromone, what you're going to see then is that signaling is mediated by a lot of proteins, one of them Fas3, which is an analog of a MAP kinase in human. And then what this leads to is proliferation. Now, our question here was, all right, we know that if you use this stimulus, you trigger this response. If you use this stimulus, you trigger this response. But we see that these proteins are also activated when you use this pheromone here. Whereas these ones here are also activated that. So how is crosstalk mediated? If they are activated and if there's crosstalk, how is it possible that you use a stress stimulus and you don't trigger proliferation, right? So these kind of questions are not well understood yet. And so we're trying to answer that. So here is how we define the experiment to try to answer that question. We used a matrix of experimental combinations of stimulus. And here, what you can see, for example, is that you you perturb your cells with salt and then measure after 0 minutes. You perturb them, measure after 1 minute, and so on. And you can do the same with the pheromone. And the tricky part to understand a little bit is the combinations of it. So for example, here you would have the experiment that you do after treating your cells, none with alpha and none with salt. But here you would have the experiments where you treated your cells for 1 minute with the alpha factor and for 1 minute with salt, and so on. In the diagonal, you will have always the same time of pre-treatment with both stimulus. In the lower diagonal, you will have the experiments where you treated the cell, for example, in that case here, let's take this one. Here you pre-treated with, or you treated with alpha for 5 minutes and with salt for 1 minute. So these different combinations of times and in pre-stimulation or in stimulation, if you will, are going to allow us to see how signaling changes depending on the time, let's say the strength of the stimulus or how long the stimulus have been there. And we're going to see that the network adapts to this, or the signaling adapts to this. Cool. So the experiment is, this is a figure from the paper. So by now you're experts in mass spec way more than me. So I'm just not going to explain to you how the experiment was done. The samples were phospho-enriched, and it's a shotgun experiment. So if you're more interested in the details, you can see it in the paper. And the first question is how to process data correctly. So what to do, for example, if you find missing values. That would be one of the first questions that you need to do before you try to look at your data to come to a conclusion. So in here, for example, you see the 36 experiments, right? Because it's a 6 by 6 matrix. And we did the experiments in triplicates. So here you have all the experiments, and here are each one of the detections. In yellow, you see when we didn't detect those peptides. Right. So how should we take into account the missing values? So one thing that we did, for example, was to remove certain peptides. I'm going to get into a bit detail as to what are the criteria to remove these certain peptides. And then, as you have seen, I guess, the machine sometimes is not able to specifically resolve if phosphorylation happens at a certain amino acid or at the one that is right next to it. So you will not, if there are two serines, for example, that are adjacent to one another, then maybe the machine is telling you with a certain confidence that phosphorylation happened in this serine, but it could also have happened on the other one. So this leads to two different phosphopeptides. Right? So you could see, for example, that sometimes the phosphorylation happened in the first case, and in some other cases it happened in the other case. And then here you would see those as two different peptides. So this is, for example, one criteria to merge them. When you know that they are so close that this can be due to resolution, then one possibility, which of course you're the experts now, so you are the ones who have to decide if you want to take that possibility, is to merge these two into one. So you just sum the intensities, you just add them, because you know that they are the same, just detected differently because of resolution. Another thing that you can do is only take those data points that are more robust. So in our case, for example, you saw that this heat map that I just showed you, in the end, was merged into 2,536 phosphopeptides, which belong or can be mapped to 1,015 unique proteins. And what we did to make it robust is that we took the data points that are shown at least in one of the 3 biological replicates and at least in 4 of the 6 salt and pheromone time points. That was our threshold to consider them robust. This is something that you need to decide in your case for your experiment. And then we use a false discovery rate lower than 1%. So again, the details are in the paper, but I just wanted to illustrate how we went then to kind of condensate, if you will, or make a data set where we know, based on our criteria, which peptides can we remove so that we don't have that many missing peptides, and so on. So here I showed you the pipeline. Now, we know which peptides we have. Do you think now I can start using this data, or is there any additional step that I need to do to the data before I can start using it?
We have the slides in front of us, so we know you have to normalize.
Damn it. Cool. So what we just did dealt with the fact that we had some missing values, but didn't touch at all these actual values. So now it's time to try to see if we can compare them. What we saw for example, in my presentation yesterday, I showed you that if you just display the raw data, because of the dynamic range, the data is going to be all over the place. You can't see anything. So one thing that you can do is take the log of the full change, and this is going to be a variance stabilizing transformation. So now all of a sudden, you're able to compare. You see these distributions. So this is each one of the experiments now, and in colors you have each one of what were the raws in the matrix that I showed before. So each one of the salt experiments, like this. Cool. So for each one of these experiments now, you're able to see the distribution. And yeah, it still shows that in some cases, these distributions are not comparable. So here we would have a problem just because this experiment seems to have a little bit of a bias here, if you know what I mean. So if you know that this is a biological effect, then you may keep your data like that. But if you want to make all of them comparable, because you know that this can be due to something in the machine, so an artifact, and that you want to correct for, then you may process all of these in the same way so that you can compare them. And so one thing that you can do, for example, is just take the z-scores. So the z-score of each value then will be the distance between each one of your data points, the mean, and divided by the standard deviation. So when you do that, what you're doing is basically trying to see how many times or how many standard deviations each one of your data points is over the mean. Yes? So it kind of tells you, basically now all of these data points here are going to have the same mean, so they are going to be centered around 0, and they are also going to be comparable because they will have the same standard deviation, 1. Cool. So because of this transformation, now I'm able to see that basically if there was value, if there were values here that were very, very high, and they were also the highest here, but much less high. Now I'm not biased because of this big difference in their absolute value, right? So I still keep the information that this was the highest value here, this was the highest value here, but now they are comparable. Yes? Good. This is just one of the many things that you can do to compare your data, to make your data comparable. Right. So are you familiar with box plots, all of you?
Just one question.
Yeah.
The colors are the experiments.
Yes. So this is each one of the experiments, but all of these are similar in the same pretreatment.
Okay.
Yes.
Yeah.
So let me. That's a good question. It's important. Sorry, I'll go back. So the first color is these experiments here. The second color is these experiments here, and so on. Go. Right. So are you familiar with box plots? Right. So, well, it's a very useful tool to see the distribution of your data, and there are some different ways or agreements to use them. So a box plot, for example, normally shows you the first quantile of your data. The second quantile is normally the median in a box plot, not the mean, which is why, actually that would be my question here to you. So I just told you that we made all of them comparable in that they all are centered around 0. So if they are all centered around 0, how come that here this line is not always at 0? And the answer is that in box plots you don't use the mean, you use the median. So what we see here really is the median. That's why it's a little bit different. But still, it's good to see that the median is similar for all of them, you see? Good. So the first quantile, the second quantile is the median, third quantile. And then what varies a little bit between the function that you use to plot your box plots is what the whiskers mean. So in some cases, the whisker is the standard deviation. In some other cases, it can be the maximum value that you have, and so on. So you just need to look at what you're using to plot box plots. But what's common is that this is the median, and that you see the first quantile, the second quantile, the third quantile, and a little bit of the deviation. So it's a good way to quickly see the distribution of your data. Good. So once you have processed your data, you have to decide what types of models are you going to use to try to understand it. So let's just see briefly an overview of the methods that we use in Julio's group. And if you have any question about some other method, I'll be happy to take it and discuss if this would be maybe better for your experiment, right? Because as I said, so here that's just an introduction. So of course I'm not going to touch upon everything that's out there. Okay, so imagine that I distribute all models in these two axes here. The first axis, the x-axis, is gonna encode or is gonna represent to the right the models that are more predictive, and to the left, on the contrary, the models that are more descriptive. And the y-axis will show how much knowledge you need on your system, about your system, to be able to use those models. So here, for example, you would have types of modeling approaches that require very fundamental knowledge about your system. So if you have, let's say, a big gene expression data set, then you don't need to know how these genes are interacting, let's say, or how the proteins that the genes encode are interacting. So you just need to know, well, there are some genes here that are involved in some process, and I want to see what's going on. Here, there would be the models used in molecular biology that require interactionist knowledge. So here you need to know that your proteins are interacting, and so on. And here, you need to know the specific mechanism that you're looking at. So, for example, not only these proteins are interacting, but protein A is phosphorylating protein B at this amino acid with a certain kinetic rate, and so forth. Good. So let's see the most common type of modeling approaches, or let's say the most powerful, depending on what you look at, of course, which are physicochemical models, such as ordinary differential equations. So these are models that require a lot of knowledge on your side to study your data. So because they are so mechanistic, you need to know specifically the reaction that you're studying, but at the same time, they are the most powerful in terms of how predictive they are. So because they are so, let's say, realistic, then they also allow you to make very realistic predictions. If you don't have that knowledge, then you can use more fundamental approaches such as principal component analysis, clustering, or linear regression. This has a massive advantage, which is that you don't need all of this information here, and you can use it then for discovery, for example. However, they don't allow you to, for example, model signaling networks. So if you want to do that, you can move on to Boolean logic, which is a more interactionist kind of approach. So there you need to know which proteins are interacting, and then you will be able to represent a network. A good example of that is what we saw yesterday in the seminar. However, it has a drawback, which is that Boolean logic is Boolean. So it only allows you to represent your measurement as on and off, or in a Boolean way, so 0 or 1. If you want to go a bit more specific and then model also or represent your measurements as between 0 and 1, so intermediate values, you can use fuzzy logic, which is a type of logic that has this advantage. However, these are not very predictive, so these are only ways to represent your data until you train them. And then you can train them, make them a bit more predictive. So this is just an overview of some methods. And as I said, some that require a lot of knowledge can be more mechanistic and very predictive, and some that require a little bit of knowledge can be very good when you want to go from discovery to validation. So this is right. So I'm not sure I made the point of how cool it is to use all of them. You can also think about using modeling as opposed to just looking at your data by eye, thinking that actually, damn it. Right, thinking that actually, well, yeah, I hope I made the point. And if you have any question about any of this, please stop me. Cool, so let's see a little bit of each one of these. So one modeling approach that you can use is networks. So a network is a model in that it represents what you know about your data set. Here you have an example. This can be downloaded from Reactome, Keck, and other databases. And they're very useful to put your data in molecular context, for example, expression data or genomic data and so on. The problems they have, though, is that they are not cell-specific and that they are not computable. So they are not models. You cannot take this network from a database and ask, well, when I use this stimulus here, what will happen here and here? You cannot compute them. You cannot simulate them, right? Another disadvantage that they have is that interactions in the databases are context dependent. So I want to make a strong point here and tell you, when you download these networks, be very careful, because all of these interactions that have been reported and put there, they are actually context dependent. So I want to be very serious about how careful you have to be. So German serious, like Achtung, like don't take them for granted. And the reason why signaling networks are context dependent are multiple. Here I review just some of them. So think about alternative splicing, for example. If you find in a database that certain proteins are interacting, but actually what you're observing is the alternative isoform of the protein that is in the database, and maybe you don't see it reacting with some other. Another mechanism that could lead to this, let's say, misunderstanding could be spatial dynamics. So think, for example, of proteins that only interact with their targets when they are in the cytosol and not in the membrane. So that's also an interaction that you have to be careful. Temporal dynamics, same thing. So some interactions only happen depending on whether the stimulation is transient or permanent. And the same goes for protein concentration. So depending on the concentrations, and this is a tricky thing because, as you know, even it's been shown that even if you take a mother cell, the daughter cells will be there will be high cell-to-cell variability in terms of the same protein that the mother cell had, how much of it is in the daughter cells. So all of these references are in the slides, and you're welcome to look at them. But anyway, I just wanted to illustrate the fact that because of these changes, then interactions that have been reported in databases may not happen when you look at your data. Okay.
But how can you check if the one you downloaded is one of these types? No.
So there are two ways to do it. One is that nowadays there are very good databases where you're able to filter. So for example, let's say that you know you're interested in a, let's say, certain tissue. So you want interactions that only happen in certain tissues. So there are plenty, well, not plenty, but there are some databases now where you're able to filter for that. Now, of course, you can also filter for organism. We're going to see that later, and so on. That's filtering. That's on the side of filtering and only taking the reactions that you know will be good in your case. Now, the other thing that you can do once you have them is use your knowledge and try to get rid of those interactions. And not be too greedy. So don't take the biggest network that you can find and assume that.
I mean,
you know.
read the paper as well.
You have to what, sorry?
Read the paper, maybe?
Yeah, that's another way to go. So for example, in the network that I showed yesterday, the one that we use in this multiple sclerosis project, it was very hard for me to find, so I retrieved a network that I started from, and then I wanted to make it as specific because of this high variability between patients that I mentioned. So because I wanted to make it so specific, then I started to look back into the papers that reported those interactions, and I got rid of all of them that were not, that didn't come, for example, from humans, that didn't come from T cells and B cells and macrophages and so on. So interactions that were found in immune-related, let's say, signaling pathways and human pathways. So that's something that you can do. Some of the databases also give you a score for these interactions. So what you can do then is try to play with that score. For example, say I only want the interactions that were found in at least 4 papers. That you can do. Cool. Another approach that you can use is data-driven approaches, such as here multiple linear regression. In this paper, what they did was to try to understand what are the differences between primary and transformed hepatocytes. And so what they wanted to see is how signaling is different between both of them. And then they take phosphoproteomic measurements from these proteins that you see marked here. And then what they wanted to do is then regress this to cytokines secreted in the same cells. So what they did is they took these measurements, these measurements, used multiple linear regression, and found how, let's say, how related those were. And how related those were are the weights that you see on the edges here. So these are related, and in transformed hepatocytes, these are not related, for example. So multiple linear regression can also be useful for your data set. Another approach that is interesting is clustering. And I want to illustrate how to use, or one way you can use clustering to answer a question that appeared to us when we were using the data set in the paper that I mentioned before. Same paper now, so same data set. Now, one problem that we had there is, how do we identify the phosphopeptides involved in signaling? So, as I mentioned yesterday, what we normally do is we start from a signaling network. But normally the signaling networks are based on proteins. What we actually measure are phosphopeptides. Can we just go and replace, somehow, put the phosphopeptides in the network so that then we will be able to use the values that we measure? Well, that's not easy. And what we did here, which was a kind of straightforward way, was use clustering to determine which phosphopeptides can we better relate to the function of the protein. And the rationale to then think, why can we use clustering of, let's say for example, from the same data set that we look at our own proteins. So here you have the proteins. Again, sorry about the resolution, maybe we can switch off the lights. But anyway, so I'll read it to you. All the way to the left, there's dig1, then there's dig2, and far1, and so on. So these are some proteins here. Now for these proteins, thank you very much. So for these proteins, this is the number of the phosphopeptides that were detected. So for this protein, dig1, we detected 7 phosphopeptides. Right, so should I go and put these 7 phosphopeptides in my signaling network, which is based on proteins? That's the question. For other proteins, we detected 3, and for some of the proteins, we detected just 1 phosphopeptide, and so on. For a protein here, we detected 9 phosphopeptides. Right, so as I said, we wanted to use clustering to determine which of the phosphopeptides are related to the function of the protein. And the way we did that is, or the rationale, the reason why we thought that was okay, even though it's a very crude approximation, and it was only a pragmatic way to deal with the data, was that within a single protein, clusters of kinase target sites can act as a functional unit. And this has been reviewed, and there are 2 reasons why this is true, or why this was shown. One of them is that it's because of the electrostatics that are regulated all together within a region of a protein. And the other one is that phosphosides can be found in clusters as well, so within proteins. So because they tend to cluster, we thought, well, maybe out of the 7 phosphopeptides, the function of the protein is driven just by one of them here, or 2 or 3 of the 7, right? So this is what we did. We clustered the phosphopeptides by their similar dynamics, and by doing that, we reduced the number of phosphopeptides to the ones that you see here. For example, for this protein, for which we measured 7 phosphopeptides, we found that out of the 7, they're acting in only 2 different ways. So you could merge the 7 into, we found that you can cluster them into 2 clusters, meaning the 7 act in 2 different ways that can be clustered. I'm going to show you this. So, for example, for this first protein all the way to the left that I just showed you, which was DIG1, then we clustered. This here, for example, is a hierarchical clustering of the same 7 phosphopeptides, and we found that they can be distributed in these 2 clusters, which are, again, what you see now here to the right. So if you concatenate the measurements of the 7 phosphopeptides for all the 36 experiments, this is how they look. This would be, for example, let's look at this blue line here. So this blue line here is the abundance of one of the phosphopeptides for the first 6 experiments, the second 6 experiments, the third 6 experiments. So I'm using the same representation as in the box plots, if you know what I mean. So this is 6 experiments, and so on. So this is concatenated. And then what you see here is that each one of the 7 phosphopeptides are, you can consider them similar, if you will, in that bunch here and in that bunch here. As you can see, this is actually very crude in this representation here, because even though these 2 peptides act similarly, like they go high here, they go low, and so on, so they are similar, they are not similar at all in all of these experiments. So this is actually just something that I had time to put together to show it to you. But in truth, what we did was not this type of clustering. In truth, we used a clustering approach which is called affinity propagation, where you get robust clusters. So instead of that, you will only get your let's say measurements clustered together if, let's put it like this, if you're confident that they are similar, let's say. So there's a way that you can be finding not only clusters but robust clusters, and to do that, one approach that I can recommend and that we can see later in detail at the hands-on, if you will, is affinity propagation clustering. So it enabled us to identify robust clusters, and it has one more advantage. For each one of the clusters, that's really cool. For each one of the clusters, it doesn't give you like an average of all of them. It gives you, for each one of the clusters, it gives you what is called the exemplar, which is the most representative of the measurement. So for example, there's a whole bunch of 5 phosphopeptides here. So this clustering approach would tell me, look, these 2 are very similar, and well, let's use this example. These 5 are very similar, and the most representative of them is one of them. It will tell me which one of these peptides is the exemplar of the cluster. Right, so that's what we used in our network. So then what we did is we looked at signaling network from databases. So we put together what we knew about this crosstalk. So we put together what regulates the response to alpha pheromone and to osmotic stress. And then what we did was we represented this as a protein signaling network. This is just a snapshot, so I cut it like this, zap. It's a part of the network. And then we went and this network, we did what, based on this clustering approach that I just showed you, we did what we could call phosphopeptide expansion. So we just expanded each one of these proteins into the peptides that we found relevant or functionally relevant by using this clustering approach. So anyway, long story short, if you have a lot of peptides and you want to use a signaling network that only contains proteins, you can cluster your peptides together and find which ones are interesting for you. Good. Another approach that you can use is it's reaction-based modeling. So this is the same that I mentioned before. It's called also physicochemical modeling. So what you do here is you use the mass action law that I'm sure you're way more familiar with it than myself, which states that the rate of a chemical reaction happening here, for example, the ligand binding to a certain receptor, is proportional to the product of the concentrations of the 2 reactants. So basically, by assuming the law of mass action and applying the law of mass balance, then you can do things like Michaelis-Menten kinetics. These equations can be represented as well, and then therefore so can be used in modeling. And therefore, this is what I meant before when I described physical-chemical modeling. So it's a very accurate way to model your data, but it has multiple parameters. So for example, to model just the binding of a ligand to a receptor, here you see, for instance, or let's say of an enzyme driving a certain reaction, you would have the enzyme binding to the substrate. This forms a complex with a certain kinetic rate, which is telling you how fast the forward reaction is happening. And then once the complex is formed, then you have a product and you have back the enzyme. So this happens. If you think about it as a network, it's a super small actually network. And even though it's so small, I already have all of these parameters here, all the forward and the backward kinetic rates of only the binding to the enzyme to the substrate. So this means that all of these parameters you can find them. So you can find how fast or what a kinetic rate is for a certain reaction. But you need to find it, and it needs to be in the same tissue that you're using and with the same conditions and so on. So this is a limitation here. If you manage to find it, or if you manage to estimate it, to learn it from your data, because you see how fast it's happening, then you can actually get it from there. Then you can also use this and make very powerful predictions. If you don't have all of this information, then you can use Boolean logic modeling, which circumvents this demand of parameters. And the way it works is that, let's assume this map or diagram or representation of this reaction here. The way the Boolean model would look is that you could represent that with an OR gate and say, either this, this will be active when this is active or when this is active. What I just said, this logic clause, let's say, can be written and used to represent this reaction here. You can also model interactions between proteins as AND gates, for example. And you can also model inhibition as opposed to activation. Right. And how do we use it? We saw it yesterday, for example. So I don't want to, unless you ask me to, I will assume that yesterday it was kind of okay, and go very briefly over it. So what we do then with logic modeling, for example, is that we assume, again, a signaling network. We then transform this into a logic model. That's what happened here, using the same notation that I just showed you. And then once all of this is transformed into a logic model, this cannot be simulated. This is just a representation of what you know or of what you found in a certain database or of what you filtered from a certain database. So to make it predictive, you still need to train it to your data, and therefore you need to acquire this experimental data. And then we're going to do a process where we're going to compare the simulations that you can do from this network to the actual data. And that's what we can see here. So again, the blue measurements are the ones that we can, so the blue nodes, using network terminology now, so these are nodes and these are edges or interactions. So the blue nodes are the ones in the columns, and they are the ones that we can measure. And this is an experiment where we didn't use neither stimulation nor inhibition. This is another experiment where we use this stimulus and this inhibitor, and so on. So upon these different experimental conditions, we can measure this data, which is shown here in black. And we can also simulate it, which is shown here in blue. Then what we do is we measure the difference between them. And when we see the difference between them, what we do is we use that to decide whether this is a good fit or not, and we display that on the background using this color that you see here. So this, for example, this darker background than this one shows that this fit between this simulation and this data is worse than this fit that is better, or this fit that is very good. Right. So from that, we can learn where our network is wrong. So upstream of those nodes that are very dissimilar from their data. And then what we can do is we can modify the network by removing interactions, for example. Meaning we can reformulate our hypothesis and restart the process where we compare it to the data. Now, this I just said again, like very manually, but actually it's an optimization problem. And what this value now that I was trying to optimize, so the background in the color, so the difference between the data that we can see and the data that we can simulate from the model, so the simulations, this distance is what we're trying to reduce. We're trying to minimize it. So it's actually a problem of optimization. It's a minimization problem. So what we want to do then is find the value that is lowest. When we actually calculate the difference between here, the difference between the data and the simulations, and we also penalize the size of the network, which is given by a relative value here and a parameter that will be computed from the network. So let's say we find a network that has 5 interactions and a network that is equally good in the score but has 4 interactions. Then this term here is going to penalize the bigger network, and we're going to consider that the final score is better in the smallest network. Okay. This principle here that I just described is called Occam's razor, as I mentioned yesterday, and it just says that among competing hypotheses, then we should always choose the one that is simplest, so the one with the fewest assumptions. Good. So this optimization problem is run automatically, so you just hit optimize, as we will see later, if you want to use it as a starting option, and can be done using heuristic approaches, such as a genetic algorithm that was the first one that we used, or that Julio actually used in his paper here. And you can also use different optimization methods. Now, using these methods here, this optimization, well, let's say this modeling approach that I just described to you was published and made open source and available, and you can find it in Bioconductor as an R package. And so you can use it programming, let's say, using your R environment, but you can also use it clicking, which is what we will do today, when you use it in a graphical environment like Cytoscape. We're going to see that in detail later. But anyway, if you want to see examples of how modeling helped a little bit to elucidate a certain biological molecular process, that's different cases to which it was applied, this approach, CellNopt. CellNopt stands for Cell Network Optimizer. So I'm being very quick here because I already described all of this in detail yesterday. If you have any question, please stop me. Yes, thank you very much.
So have you ever tried? So this you make all of this computation based on some experimental data that you still have chosen.
Yes.
Somehow, no, you have a filter. Have you tried to do exactly the same with a different filtering of your data? You know what I mean?
Yeah, that's a very good question.
The bibliographic one, I say, at least. Not only your base,
yes,
your database, but also the bibliographic data that you base everything.
The bibliographic data is, sorry.
You say you filter depending on, so I only to believe the interaction that happen in the same tissue.
Yes.
This kind of thing.
Yes.
So changes this filtering. Have you ever tried to repeat it? It's a lot of work. Maybe this is stupid what I say, isn't it?
No, it's a very good question. So it goes back to what he was asking before. Yes, you can assume a signaling network and compare it to your data and try to find what's the network that best fits the data. And then you can restart the whole process starting from a different place. For example, from a network where you only took the interactions that were found in a certain tissue.
And you used to find a lot of difference?
Yes, it's very different, of course. So the hard thing when you're using this approach is that this optimization, because of this assumption of Occam's razor, the only thing you can do is assume all the interactions that you want from the literature and remove them to see if when you remove them, this is more similar to your data. But you can only remove, you cannot add interactions. And this is what's done during the optimization process. You start removing, comparing every time against the data. And then of course, if you start from a bigger network, then you will be able to, you have more chances to find one that will fit better by removing, right? You have more to remove. But you can also be introducing false positives. So you will end up finding a network that fits very well your data that has nothing to do with what's going on in your biological experiment. That's why I would recommend to be very stringent when you use the signaling network at the beginning. Don't just download the whole human interactome and then compare it against your data. If this would be then what you can call And so it's a problem where you have so many data points compared to all of your variables that you will always be able to find the solution. Yeah. So be very stringent when you select your data and your network, and try to make it as specific as possible. And always take into account how many data points or signals, how many measurements you have. So if you have a network like this, here, so you're going to be very confident about, for example, the behavior of this node, which is in between these two. So if you learn that this should be there because it's actually very important according to your data, then you're going to be able to make a safe conclusion about that because it's in between two measurements of yours. This is just as a rule of thumb, and there's plenty that we can do here, and we will do it later when we see this. So this is the identifiability analysis that I mentioned yesterday, and that we're going to do later in detail. Yes?
When you look to this plot,
Yeah.
the ones that are more in yellow are the ones that are not flat. So the flat, I don't know what the axis of these plots down there.
Mm-hmm. So the axis here?
These ones, yeah.
Yeah, so this is each one of the time points where you're measuring, in that case, for example, the behavior of RAF1. Yeah. And this is the intensity normalized between 0 and 1 of RAF1.
To change or not to change?
And you say what, sorry?
You expect them to change over time, or?
Depends on the stimulation, that's a good question.
The ones that change are the ones that are not confident, not the ones that have some more color, in this case.
That's a good thing to look at, but it shouldn't happen. So for example, let's say RAF1. RAF1 should be active when you give a growth factor, right? So because it's a MAP kinase and it's mediating proliferation. So you should see that it's first going up and then goes down. And then when you use, for example, EGF, which is here, that's why it's going up, because you're using this growth factor. Then it's going down, and this can be modeled because we are actually finding this path in the network. So in the network, we are representing that when EGF is there, this should be active, then this should be active, then this should be active, then this should be active. So what the simulation is going to do is, if EGF is there, this is going to be active. Right, that's why it goes up. And the reason, sorry, yes. So what is a bit confusing here is that I have been talking about Boolean modeling the whole time, but actually what you have here is a bunch of time points. This goes up and goes down. This is not Boolean at all. So it has, well, it is Boolean in that it goes from either to 0, from 0 to 1 and back to 0. So it's Boolean. But what I think you're worried about is that you think that the models are going to be worse, so you have a darker background when there's something happening. That's what you're saying. That shouldn't be the case. So for example, if you look here, you see a very good fit, even though GSK3 was very high in this experimental condition. And then it went down, and the model correctly predicts that because of the structure in the network.
Yeah, I was more worried about the ones that go up and then down than the ones that just go down or just flat.
Well, I mean, there's also something that is called the residual error, which is the error that there's going to be here anyway, just because using a Boolean approach, you're not able to capture very subtle changes. So actually, the model is correctly telling you that based on this, when you give EGF, this should go up, this is doing it, and after a while it should go down because of this negative feedback loop. So when this is happening, so the simulation is anyway doing that. You still see a darker background because it's not doing it at these measurements here. Some of these differences, though, are just residual. So the model is never going to be able to say, well, I should go from 0 to 0.8 because it's Boolean. It can only go from 0 to 1. So yes, it's true that this is a bit more difficult to model than all zeros or ones. So here you're going to be, just intrinsically, because the model is Boolean, you're going to be able to capture this, to simulate this better. You're right. But still, I would consider that a good model. It's just indicating that that there is a bit of difference just because you're not able to represent a 0.8, which is this here. Right, that's a good question. Thank you very much. We're going to see later, for example, the cases in which you assume a totally wrong network. In that case, you would see a red background here, telling you that, yeah, the data is going up and down, and actually, based on the network that you gave me, I can only simulate that it's staying flat. So we're going to see how to infer that the assumption you made, your hypothesis, is completely wrong, and how to change it using an educated guess. Cool. So, finally, the last slide about CellNet Optimizer is going to be that this actually goes back to your question. Depending on the type of data, you may want to use the different mathematical formalisms. You just saw, thanks to this good question, that there's a limitation in using a Boolean approach. So you assume that your values are between 0 and 1. Sorry, not between 0 and 1, either 0 or 1. However, this has a really cool advantage, which is that you can use it when you have very few, it doesn't have a lot of parameters. Whereas, as I mentioned before, the physicochemical modeling has a lot more, or requires a lot more parameters, thereby a lot more knowledge on your side. So there are a bunch of different approaches drawn here that you can use in this modeling approach, in this CellNopt. CellNopt-R is the first one that was published. It's the one in the paper that I mentioned before, and it's the one in the diagrams that I have been showing. It's also the one that I use in my research in this multiple sclerosis project that we looked at yesterday. So when you have, let's say, scarce data, or when you have a small amount of measurements compared to how big your network is, then, and these partial effects that you just asked, so these intermediate values are negligible, or you're not interested in seeing these specific simulations that go exactly to 0.8 in the slide before, then you might use CellNopt-R. If you want to use, if on the contrary, you want to use an approach that is able to model intermediate values, then you might use CellNopt-R in the fuzzy logic version. If you want to simulate 2 time points, you might use CellNopt-R 2 time points, which is just an adaptation, an extension of this mathematical approach to be able to use 2 time points instead of just 1. If your data is very rich, so if you have a lot of measurements, then you're able to look at dynamics. When you do that, then you can use 2 different approaches. One is CellNopt-R dt, and the other one, so for discrete time point, and which is just an extension of this again, or CellNopt-R, which is based on ordinary differential equations. These are logic ordinary differential equations, and because of what I said before, they are able to model these dynamics. It's a good case to use them in this case here, because then you're able to see the dynamics when you have a rich data set with a lot of time points. So again, you have a lot of time points, you want to use an approach that is able to represent dynamics. You don't have a lot of time points and your network is very big. You want to use a simpler approach. And these are all R packages that you can use if you are interested in using R, or you can use them graphically, but that's a bit tricky. So we're going to see it, later that it gets a bit complicated to use all of this graphically, just because you want to be able to change a lot of things, and graphically is a bit difficult to do that. So this one here is a good case though, but that you can use just clicking and in a graphical environment, and we're going to do that later in the hands-on session. So I would like to say thanks to everyone in the Julio's group who will be now moving to Aachen, as I described yesterday. So this, what I showed today, is based on work that we have been doing during the past 2.5 years. And now the group will be moving to Aachen, and I will not be any longer in EBI, unfortunately. And yeah, so this is I think all the theory that we need. So now we're going to go hands-on. And I hope you're familiar with PhD comics. They are really cool, of course. So yeah, the hope is that this is not going to happen, because if it does happen, I will start answering just randomly that the answer is pi over